In this chapter, we're going to look at the genome and the biochemistry of it. It's chapter 10. Just very generally, history of genetics, microorganisms. They were used to study genetics because they grow fast. It's easy to get the DNA separated out from everything else in the cell, with especially bacteria. So from a historical standpoint, microorganisms were used to determine, there were lots of different experiments done, but to finally determine that yes, it's DNA that's responsible for heredity that contains the genetic material. There's a classic study where it was done, they finally were like, is it DNA that's responsible for heredity and the genetic material, or is it protein? And it was finally determined that it was indeed the DNA. In 1953, Watson and Crick proposed the structure that DNA was indeed a double helix. Now, you may read places where Franklin was also involved with this. She did a lot of X-ray crystallography work on it. Watson and Crick were eventually awarded the Nobel Prize for determining the structure of DNA. And depending on what book you read, it tends to cycle up and down. Sometimes they make a big deal of it, and then it kind of goes away, and then later it comes back again. But why did Franklin not receive the Nobel Prize? Because Watson and Crick could not have figured out that it was a double helix without her work. That part is pretty well established and true. So you can argue, should they have mentioned more in their papers and recognized her more for her work? You can argue, yes, they probably should have. Did she get the Nobel Prize? No, she did not, because until just a few years ago, Nobel Prize was not awarded to anyone who had already passed away. The idea was that it was to recognize you for outstanding, groundbreaking work in a field, and that you were continuing to work in that field. Unfortunately, by the time Watson and Crick were awarded the Nobel Prize, Rosalind Franklin had already passed away. And so, like I said, that was, at that time, because she passed away, it was never given to someone who had already passed. So that's why she didn't get it. I actually remember the first time they did award posthumously a Nobel Prize to someone. It was probably about 10 years ago, and it was quite the controversy that they were announcing publicly who the winners of the Nobel Prize were for that year. It was actually in medicine, and it was on a Monday. And the previous Friday, this one researcher passed away. He had spent his life doing research with cancer, and in a sad, ironic twist, he actually died of cancer the Friday before they announced the winners of the Nobel Prize. He did not know when he passed that they had selected him to be the recipient for the Nobel Prize in Medicine that particular year. He was not aware of that. So he died literally 3 days before they were to make the announcement. And so they had everything prepared, ready, to announce that he was the winner. And then he passed away. And that was a huge, huge debate. Oh my goodness, I can remember it being all over the news. Some people were saying, we've never given it to someone who has passed, and he passed away, and we're really sorry, but we need to go back and decide now who's going to get this award, because we've never given it to someone who had passed away. And then other people were saying, my goodness, literally 3 days. Come on, just give it to him. And it was quite the argument. They finally did award it to him in his name. I believe it was like his wife and son or something accepted it in his name on his behalf. But sometimes you'll read arguments about Rosalind Franklin, about why didn't she receive it. Well, she had passed away at that time. But it was in 1953 that they did the groundbreaking work to determine that, yes, DNA is a double helix. In 61, Jacob and Monod proposed existence of RNA, the second type of nucleic acid. In the 1990s, the genome sequencing projects were beginning. In 1997, E. coli genome was finally completely sequenced. Bacterial genomes are smaller than, say, human genomes, so we start with the easiest first. Finally, in 2003, the human genome sequence was completed. So some of this is relatively recent, relatively new. The DNA structure is usually double-stranded. Notice I will say usually, some books will say always, just because there are some viruses that have single-stranded DNA. The monomer is of the nucleic acid is what's known as a nucleotide. It's composed of a sugar, a phosphate, and a nitrogenous base. The sugar is going to be deoxyribose. These nitrogenous bases, they get their name because they have nitrogen, they're ring structures. There's 4 of them: adenine, cytosine, guanine, and thymine. Just by tradition, they are always abbreviated with the first letter, and it is always capitalized. So in this picture, it is showing the nucleotide, and that you've got the phosphate group, the sugars in the middle, and then the base on the other end. Deoxyribose is a 5-carbon sugar. 4 of those carbons are on the ring structure, and then 1 carbon is off of the ring structure. So when we look at those nitrogenous bases, there's 4 of them. They're classified as either purines or pyrimidines. The purines are double ring structures. Pyrimidines are single ring structures. The purines, this will be your adenine and your guanine. Some people remember it as pure as gold. Purine, A for as, G for gold. The pyrimidine will be your cytosine and your thymine. Now you do not need to know the structures as far as being able to draw them out, but here you can see that the pyrimidines are the single ring structure, and then over here your purines are the double ring structures. So this DNA, finding out that it was a double helix was a huge, huge thing. The way to set up what you would refer to as like the backbone, and the backbone, it's kind of like if you visualize a ladder, it would be the sides of the ladder that you hang on to as you're climbing onto it. That's going to be alternating phosphate, sugar, phosphate, sugar, phosphate, sugar. The nitrogen bases, those are going to be pointing inward. And because it's a double strand, the nitrogenous base from one strand is going to hydrogen bond to the complementary nitrogenous base on the second opposing strand. So it's kind of like with the image of a ladder, that backbone, phosphate, sugar, and off of the sugar is where the nitrogenous base is, pointing inward. That's forming the rungs of the ladder. You know, somebody asked me one time when I said rungs, they didn't know what I was talking about. It's where you put your feet as you climb the ladder. And so one nitrogenous base, hydrogen bonds to the complementary nitrogenous base on the complementary or the other strand. Now, there is a certain way that these nitrogenous bonds will pair. Adenine and thymine will always pair together, and they are going to be held together by 2 hydrogen bonds. Guanine and cytosine will always bind together with 3 hydrogen bonds. Different ways of remembering this. I one time had a student who said the way she remembered it is that there are 2 apple trees. And then she said the way she remembered the other one was that everybody wishes they had a 3-car garage. But there's many different ways you can remember it, as long as you know that A and T bind with 2 hydrogen bonds, G and C bind with 3. The strands are what we call anti-parallel. There's a way of looking at it with the way the sugar is oriented, where they're going in different directions. This, we don't spend a whole lot of time with it in here because we just frankly don't have time, and it involves more chemistry as far as looking at the orientation of the sugar. I'm just going to say this, it makes a difference in the way the strands are running, that you know they're anti-parallel, because it's going to come and play a role when we talk in the next chapter about DNA replication. The DNA contains all of the genetic information. Everything about it is there. And so this is just showing once again that you have to visualize this. You have a phosphate group, you have the sugar. The phosphate group, the sugar. The phosphate group, the sugar. This is your backbone here. And then off the sugar, you have your nitrogenous bases here. So it's like you're forming that ladder. This is showing the X-ray pattern, that you can see that it is indeed this double helix. So you can, because of the helical shape, you can basically see here and then here the way the orientation of it is going to be. So in this picture, I think it's a lot easier to see than the previous one. The previous one, it was as though you were looking down on this. So here you've got the 2 strands. Like I said, it's a double helix. It's twisted around. We know exactly the distance between each turn, so we know everything about it now. But you can see here you've got 1 strand here, then complementary strand that's opposite it, and you can look through here at the bases that you have A binding with T, G binding with C, and just if you know the base sequence on one strand, you can figure out what the base sequence should be on the complementary strand. When I say a complementary strand, I mean the other strand, the other side of it that's bound to it. And in here you can see these dotted dash lines. That indicates hydrogen bonds. And so you can see there's 2 sets here between the A and T, and then there are 3 sets between the G and C. This is just another way of showing it once again, the hydrogen bonds that are holding the basis of the complementary strands together. It's very important that those are hydrogen bonds, which are the weakest of bonds, because whenever you go to make a protein, you are going to have to separate those 2 strands. It's kind of like a zipper. You're going to have to unzip it. So you're going to have to break these bonds. You want to have bonds that are fairly weak, so it doesn't take as much energy to break them. Now, the bonds between the sugars and the phosphates over here on the side, all the bonds here along the backbone are covalent bonds. You want those bonds to be very, very strong, because you do not want these backbones to break. If they break, you are in deep trouble. But you need to break these bonds here, the hydrogen ones, in order to make proteins to separate those 2 strands. So when you think about that, the cell, number one, doesn't want to expend extra energy if it doesn't have to. You can say it's either lazy or it's efficient. You know, don't make me work if I don't have to. So you put the weakest bond where you're going to be breaking the strands apart a lot. It starts to make sense when you put it in those terms. Now, the other nucleic acids are RNA. RNA is usually single-stranded, except in viruses. You may have some that are double-stranded. Once again, the monomer is a nucleotide, so it's a sugar, a phosphate, and nitrogenous base. The sugar, however, for RNA, ribonucleic acid, is going to be the sugar ribose. The bases, those nitrogenous bases, once again, will be adenine, cytosine, guanine. However, now it's going to be uracil for the 4th one, not thymine. Thymine is found only in DNA. Uracil is found only in RNA. And once again, you use the capital letters for RNA just like you do in the DNA when listing the bases. So some of the differences here, over here on the left, this first compound is the deoxyribose. That is what is found in DNA. And then ribose is the sugar that's found in RNA. So both of these are sugars. The only difference between them is right here. On the DNA, that is just a hydrogen. But on the RNA, there's an OH. So that's why deoxy, you took the oxygen off. That's the only difference between them. And then over here, you've got the thymine versus the uracil. DNA, like I said, is usually double-stranded versus RNA that is usually single-stranded. Now, even though RNA is usually single-stranded, it usually kind of folds up on itself for a 3D structure. So the DNA contains all the genetic information. RNA is going to be involved with protein synthesis. It's going to take information from DNA and then use that information to help make proteins. There's several different types of RNA. There's 3 main ones that we talk about. There's messenger RNA. We abbreviate that as mRNA. What it is going to do is it will use DNA as a template, copy the information from the DNA, and use that for making proteins. Ribosomal RNA, which is referred to as rRNA, this becomes part of a structural component of the ribosomes. And you remember the ribosomes, that's where the protein synthesis actually occurs, the site of it. Transfer RNA or tRNA, this is going to bind to amino acids. It's going to be, there are at least 20 different types of tRNAs because each one is very specific for which amino acid it can bind. So if it can bind the amino acid alanine, that's the only amino acid it can bind. So it's very specific which amino acid it will bind, and then it transports it. The amino acids are just floating around in the cell. The tRNA will go bind to the specific amino acid and then transport it or bring it over to the ribosome when needed for making the new proteins. We will get into the next chapter as to how protein synthesis, which is known as translation, how this actually occurs. And so you're going to have your ribosomal RNA that's part of the ribosome, which is shown here as these 2 different structures. You've got your mRNA that is this long strand here. Where did it get the information from? It got it from the DNA, and that contains the information on how to make protein, which is what you are making, the strand up here. Your tRNA is bringing in the appropriate amino acids. mRNA is here, and the rRNA is part of that ribosomal structure. This shows what a tRNA looks like. It's often described as kind of a clover leaf. So way down here on the bottom, here is your messenger RNA or your mRNA. And then here, this structure right here is the tRNA. Here's your amino acid up here. That binds on the end away from where it's going to bind to the mRNA at the opposite end. And then there's an area here that will bind, the bases will be complementary, and then will bind to the site on the mRNA. Like I said, we'll get into the steps of how all that will work in the next chapter. The genome, you're talking about the entire genetic material. What's all that information there? Genes are just small segments of the DNA. You have literally thousands of genes that control everything. As humans, everything about us. Same thing with bacteria and fungi. It controls everything about it. Genotype, this is the collection of genes that are on the DNA. Probably one of the best ways of explaining this is that as humans, we have 2 copies of every gene. So let's say for eye color, you might have a gene for brown eyes and a gene for blue eyes. That's what's on your DNA. That's the genotype. Phenotype is what you're going to physically see. And now that's going to be determined by the genotype, by the genes that you have. Environmental conditions may play a role in that. So for humans, if you have one gene for blue eyes and one gene for brown eyes, that's your genotype. Phenotype is brown eyes. You're going to have brown eyes. That's what you're going to look like. Now, in bacteria, oftentimes you only have one copy of a gene. But if you have a gene for a pigment, that gene, that's your genotype. That's what's on the DNA. What do you actually see? Well, there's a bacteria we work with a lot in the lab that when you grow it at room temperature, it's going to look red. That's the phenotype. Now, when I say it may be influenced by environmental conditions, you grow that same bacteria at 37 degrees C, which is 98.6 Fahrenheit. Now it's going to look kind of a beige, whitish color. The chromosome, this is referring to DNA that's containing most of your genes. So we do have a lot of different terminology that we use for this. Now, this is the bacteria I was talking about, Serratia marcescens. This is what we use in our lab. So like I said, at room temperature, it has that expression of the red pigment gene. That gene is turned on, so it's going to look red. However, at higher temperature, it's like it's turned off. So when we say a gene is being expressed, it means that it's turned on. You're going to see the phenotype of it. Same organism here. So the DNA in prokaryotes, it's haploid, meaning you only have one copy of every gene, and it is circular. In eukaryotes, like fungi, it's linear, and it's diploid, meaning you have two copies of everything. Non-coding DNA refers to DNA that does not code for proteins. It's often containing regulatory information and helps to stabilize the DNA. Sometimes you have extrachromosomal DNA in prokaryotes. This is usually plasmids that may contain things like drug resistance. In eukaryotes, extrachromosomal DNA, we're talking about the DNA that's found in the chloroplast and in the mitochondria. And so this is showing that in chromosomes you have usually a fair amount of DNA that's what we call the non-coding. So on this diagram it's showing that the actual genes are just kind of this color right here, but you have this extra material in between that doesn't really code for anything. It's just extra. There's a lot extra with the cell. In terms of how big is the genome when you compare different organisms, there's a huge variety. And sometimes it kind of depends on, if you're comparing two things, how close are they to each other as to the size of it. This talks about how many base pairs are there. In humans, it's 3 times 10 to the 9. So you write a 3 and you put 9 zeros after that. So 9 billion. Mammals, which obviously humans are mammals, so we're going to be in this area. This shows the ranges of some of your other eukaryotes like plants and other animals. Fungi are down here, 1.2 times 10 to the 7th. E. coli is smaller. It's a bacteria. Viruses are much, much smaller. Here's an example of 2 viruses. So there is a wide variety in the size of the genome.